Thank you, and this was massively impressive, as you promised. And first, I would like to start to thank the organizers for putting on this conference and, of course, for having me. And I think in the spirit of Sophian, my talk has two parts. First, I will talk about the development of a new technology that we did in my lab, and then show you how we used this technology to learn something new about the biology of force-gated piezo ion channels. So I think it's fair to say that every lab that studies force-gated ion channels is using the following technique to study them, and that is pressure clamp electrophysiology. Just to give you a brief introduction, so in this technology, you perform a cell-attached patch on a cell that expresses your force-gated ion channel of interest, and then you can apply negative pressure or suction in the patch pipette here through a pressure regulator. And that will bend the membrane here and create tension in the membrane and then activate the force-gated ion channel. So similar to a researcher who would use a voltage step protocol to test a voltage-gated ion channel, you can then use this technology to apply increasing steps of pressure here in a pressure step protocol. And if you do this on a cell that expresses the force-gated ion channel piezo 1, you can then observe currents that look like this, where as you increase the negative pressure, you obtain larger and larger currents that then inactivate over time. That's an inherent property of piezo ion channels. So this technique is incredibly useful. You can learn something about the channel number in your patch, inactivation kinetics, and of course, the pressure sensitivity. But crucially, there's one big disadvantage of this technique, and that is that, of course, force-gated ion channels are not directly sensing pressure. They're activated by tension in the membrane. And so in this assay, what you do not know is actually what the activating stimulus was. There is a technique that can get around this, and that is by combining this technique with DIC, differential internal contrast imaging here, where at the same time you use a camera and you image the patch at very high magnification. So when you do this at 400x, you get an image that looks like this. Here, these black bars are actually the outlines of the patch pipette. And then you can see here this optical density is actually the membrane. So in fact, it's actually not the membrane because lipids don't diffract light very well. It's the underlying cytoskeleton. But what you can do with an image like this is then perform a circular fit to obtain the global radius of curvature of the membrane. And because you know what pressure you applied to the patch pipette, you can then use Laplace's law to calculate the global membrane tension. And with this, you can obtain a tension response curve. And that is, of course, important because that truly characterizes your ion channel, and you can learn something about the sensitivity and dynamic range of the ion channel that you study. In addition, these tension sensitivity curves are needed to calculate the biophysical parameters of channel function for these channels. So this is, for example, the Gibbs free energy of gating and the delta A, the area expansion that the channel undergoes when tension is applied to the membrane. And these parameters are important because then you can use them to compare your data to very different other techniques such as atomic force microscopy or reconstituted methods like giant unilaminar vesicles. Okay, so this technique is good. We in our lab have used it as well, but the downside is you don't actually have direct control over tension. You just analyze it post hoc in the assay. And also this image analysis is incredibly hands-on and cumbersome. So in order to overcome this, we've created a method that we call optically guided tension clamp, or OGTC. And basically, this method has 3 components to it. It consists of an image analysis program that is completely automated, a dynamic tension control program. Both of these are software units. And then a hardware unit that is basically just a microcircuit board that connects all of these components so they can work together. When the system is in action, it looks something like this. So this is in real time, basically a protocol that we are running here. And you can see in red, you can see the fit here on the membrane. The way this works is that we trained an AI machine learning algorithm with 1,500 images that we had previously acquired in our lab of patch pipettes containing membranes to automatically recognize the membrane density and then fit it. With a dedicated CPU, the algorithm can do this in only about 4 milliseconds. So this is then fast enough, we thought, to basically run this analysis in a closed loop system so that we can adjust the pressure in the patch pipette as we go in order to achieve and effectively clamp membrane tension to any value we want. Just to characterize the system performance, you can see here when we apply a tension step from 0 to 5 millinewton per meter, you can see here in red and blue the curvature of the membrane and the pressure actually fluctuate and change over time. But after only short equilibration, the tension here hones in on the target value of 5 millinewton per meter. Below here, you can actually see a full tension step protocol executed, where each time we step from 0 to 1, 2, 3, 4, 5, 6, 7, 8 millinewton per meter. The system performs quite well. The accuracy is greater than 93% for low target tension values of 1 to 3 millinewton per meter, and better than 97% for high tension values of 4 and above. And the precision of tension, so the fluctuation around the mean, is also quite good. It's better than 0.3 millinewton per meter. The response time of the system is relatively slow still. It's 400 milliseconds for low tension steps of 1, 2, and 3 millinewton per meter, and 1.5 seconds for high tension steps. But crucially, after only a short equilibration here, there's virtually no drift in the system. So what this then allows us to do is actually to measure tension response curves of ion channels that either express at very low levels or that have a very low conductance, or both, simply by averaging the current response over time. And this you're not able to do with the method that I showed initially. So one channel that fits this profile are TMM63 channels. So here you see measurements of TMM63A, here a tension response profile, and here typical currents of TMM63A. And this is in cells that have been CRISPR engineered to lack Piezo 1 ion channels, and these cells are completely mechanically inert, so the currents here are all coming from TMM63A. Here you can see the tension response curve of a single measurement, and here aggregated over several measurements. So this now, for the first time, we have learned that TMM63A actually responds to membrane tension with a T50, a tension of half maximal activation of about 5.5 millinewton per meter, and it has a relatively shallow and gradual response curve. So it's able to transduce actually tension values or tensions from very low tensions to relatively high tensions. So in fact, this tension response curve is relatively comparable to those of K2P potassium channels, track 1 and track. They have a very similar profile, but this profile is very different from, for example, MSCS and MSCL channels. These are bacterial force-gated ion channels that only open near lytic tension of about 10 millinewton per meter. And the response curve is also very different from Piezo 1, which is the most sensitive force-gated ion channel that we know of, that has a T50 of only 1.4 millinewton per meter. So this suggests that these channels are very good at gradually transducing various tensions over a very wide range. And finally, a last validation, we wanted to test our favorite ion channel, Piezo 1, and without going through the data, we were able to fully recapitulate the measurements we had done previously with the older method on Piezo 1. Now, and this brings me to the second part of my talk, which are piezo ion channels. So piezo ion channels transduce various forces throughout our bodies and have many different physiological functions. Importantly, there are two different piezo ion channels in your bodies. Piezo 1 is relatively widely expressed. Almost every cell type in the human body expresses piezo 1 ion channels. And it senses more internal forces, for example, substrate stiffness, and is important for cell migration, cell division, cell proliferation. Piezo 2 is the channel that is, in its expression profile, more limited to neuronal tissue. So it's expressed, for example, in DRG neurons, proprioceptive neurons, and the vagus nerve. And there it functions, for example, as a sensor of light touch, proprioception, lung inflation, and so on. Now, one thing that has bothered me actually since piezos were originally cloned, which is now 15 years ago, is that there is still no satisfying explanation for why there are actually two different piezo ion channels that are needed. First of all, and that was a big surprise when we heard about it, the structures of Piezo 1 and Piezo 2 are virtually identical. So this is an overlay of three different structures of Piezo 1 from the McKinnon lab, from Bailong Zhao, and from Andrew Ward's lab, and a structure of Piezo 2, and you can see that they match almost perfectly. Also, in terms of function, both of these channels are virtually identical. There is a difference in the kinetics of inactivation. So Piezo 1 is a channel that inactivates with a typical time course of about 30 milliseconds. And Piezo 2 inactivates with a time course of about 10 milliseconds. So there's some difference, but this inactivation kinetics is highly plastic. It can be modulated by extracellular pH, by voltage, by lipids. So for me, it never really made sense that just because of this, you need two different proteins. So now with this tool, when we had this in hand, of course, what we tried and were asking is, do they differ in their tension sensitivities? And lo and behold, they do. You can see this here in the two different sensitivity curves that we have recorded. They are actually different. This can be better appreciated when you don't plot the tension response curves, but actually the tension sensitivity, which is the derivative of both of these curves. So here you can see now that both channels are actually different in their midpoint. So human Piezo 2 is the more sensitive of both channels. They differ almost twofold in their midpoint. And in addition, you can see that human Piezo 2 here has a higher response, meaning it has a steeper response curve. So it's more switch-like, versus human Piezo 2 has a more broader range and actually transduces tensions of up to 7 millinewton per meter. But the differences don't stop there. Namely, it turns out that Piezo 2 is heavily spliced in a cell type and tissue-specific manner. And this goes back to work from Mark Kuhn's lab, who is at the NIH, who published about 8 years ago now sequencing data showing that Piezo 2 is spliced and has 7 different exons that are spliced, again, in a cell type and tissue-specific manner. And of course, that implies that there might be a difference in function that is conveyed by the splicing. So that's our hypothesis that we followed up. How does the splicing look like? So this is a topology plot of Piezo. And you can see actually that 6 of these spliced domains are all in intracellular loops, One of them, and I'll point this out later and come up to this again, is actually in a transmembrane domain. So this is a strange place for splicing, because if this is indeed spliced, you would expect that the entire topology orientation is changed, and probably the channel would be non-functional. Interestingly, there's absolutely no structural information on these domains. So in the structural data that I'd shown you before, none of these loops are actually resolved. And this implies that these loops are probably highly flexible and therefore not resolved in cryo-EM structures. In addition, AlphaFold completely fails to assign any structure to these loops. So in order to now study the functional effects of splicing, we decided to go for a purely biophysical structure-function approach. And first of all, we generated two piezo constructs, one that we call piezo 2-min, which is basically the piezo protein, but now this time lacking all splice domains. And then another variant that we call piezo 2max that includes all of these domains. And then we recorded tension sensitivity curves of both of these variants. And now you can see actually both of them vary really drastically in their tension responses. Piezo 2max is the more sensitive one, so this is the one that contains all spliced axons. It's almost as sensitive as piezo 1, and it's a very steep response curve. So this really almost functions like a switch for membrane tension sensing. Piezo 2-min, however, is much more broadly tuned and less steep. Okay, how does each of these splice domains contribute to this difference? In order to learn this, we made 14 more constructs. 7 were we introduced into piezo 2-min, one splice domain at a time, and measured its function. And then the reverse were from piezo 2max. We subtracted one domain at a time. I'm not showing you all the data, but just the most important highlights. And it's a busy slide, but I'll walk you through the data. You can see here that for when we subtract splice domain 19, 33, and 35, each time you lose sensitivity to membrane tension. So from these data, we conclude that domains 19, 33, and 35 are each individually necessary for maximal tension sensitivity. When you delete splice domain 40, there's absolutely no difference here. So maybe it does not contribute to tension sensitivity, but has some other function. One idea that we have is that it might actually control membrane expression levels or location, and we're now following actively up on that. And interestingly, the splice domain 22, which I pointed out is actually a transmembrane domain. When you mess with this, it leads to non-functional channels that just don't even make it to the membrane. So we think this might actually be an error in the sequencing data, and we're mining currently long-read sequencing data to understand this better. In addition, and that's my last point, there's a special role for exon 33. Bailong Zhao had already shown a number of years ago that exon 33, which is actually the single domain that is spliced in piezo 1, controls the conductance of the ion channel. And there they had beautiful structural data where they actually showed that exon 33 forms an obstruction in the outlet of the ion permeation pathway. So when and he called this domain piezo 1.1. So when you delete this, you end up with a piezo ion channel that has a substantially higher unitary conductance. So first we wanted to see if this actually also holds up in human piezo ion channels, because his original study was done in mouse. So this is our data, and you can see this effect recapitulates. And importantly, this also is exactly the case in human piezo 2. So I'm not going to walk you through the individual data, just the summary here. So you can see when you add domain 33 to human piezo 2, you end up with the low conductance of piezo 2 max, but when you delete it, you end up with the high conductance of piezo 2 min. So this shows that exon 33 is both necessary and sufficient to determine unitary conductance. So it plays a dual role in tension sensitivity and conductance. So finally, how might this work actually mechanistically? So for this, you have to know that piezo 2 is a highly curved membrane protein, and it's thought to actually bend the membrane that it's sitting in far beyond the extent of the protein in what is called the piezo footprint. However, under tension, the protein can actually adopt a flat state that is almost completely flat. And this looks like this. So if I flick back and forth a little bit, you can sort of imagine how membrane tension can pull on this protein and induce this flattening. A number of years ago, Rod McKinnon, together with Christoph Haselwanter, has actually shown first in theoretical work, but then also with experiments, that it is the mechanical rigidity of these so-called piezo blades that competes energetically with the membrane bending stiffness and the energy from membrane tension. So in other words, the stiffer the blades here of the piezo protein are, the higher its sensitivity to membrane tension is. So and this sort of provides an idea or rationale for how the splicing actually can change membrane sensitivity. All the splice domains are likely located here somewhere, so it's easy to imagine how adding and subtracting them can alter the rigidity of the piezo blades. Of course, this still needs to be tested. Okay, let me briefly sum up my talk. So first, about this optically guided tension clamp, I want to briefly discuss some limitations. So first of all, one limitation is that the microcontroller we developed is only for Hika amplifiers. It should also work for Axon, but we haven't tested this yet. The speed of the system is still too slow to measure rapidly in activating ion channels like piezo 1. But with a faster camera that can actually capture images in about 10 milliseconds, you should be able to get up to the speed in order to do this. And lastly, because you need to image the membrane inside a patch pipette, this only works best when you have really high contrast images, and you need to use relatively large pipettes of 1.5 to 2.5 mega ohm, and this can sometimes limit the throughput of the system. This being said, overall, you achieve a throughput that is almost as high as regular cell-attached patch clamp electrophysiology. The system is completely automated. I didn't talk about this, but all the error analysis, error propagation is all built into the system. So it's as close as you can get to an unbiased tension clamp. In addition, we can imagine that the system could be used alternatively as a curvature clamp to study how proteins that either induce or stabilize membrane curvature associate with the membrane. And for the second part of my talk, I've shown you how piezo 2 splicing determines tension sensitivity and conductance. The limitation here is that all of this was now done in overexpression systems. We still want to test the physiological validity of this by using primary cells to really check if our findings hold up. And of course, the mechanism that I just proposed to you for how tension sensitivity is changed is only speculative. But with the actual data that we have generated, other methods like AFM and GOV measurements can actually now test if this is the case and if splicing changes the stiffness of the protein. The rationale or the significance, of this is, of course, it provides a rationale for why you would need 2 piezo ion channels and for why this cell type specific splicing exists. But more excitingly, I think, and that's my last point, is that now we have, for the first time, targets in the piezo protein that we think are good domains for changing its function, its sensitivity in a cell type specific way. So I think it would be really nice to target these intracellular domains with small molecules. Okay, and then I have to thank my graduate student, Michael Sandoni. All the work that I've shown you was done by a single graduate student. Michael, he's now on the job market. If you need a talented postdoc, come and let me know. All of this work was performed at Duke University, and these are our funding sources that support us. Thank you.
Thank you, Professor Grandl. That was beautiful stuff. Any questions?
Thank you very much for interesting story. We have developed tension clamp using lipid bilayers. The lipid bilayer is very simple system, and the relationship between pressure, tension, and curvature is straightforward.
Yeah.
Compared to the lipid bilayer, patch clamp is very complex system. So is there any sign that the cytoskeleton affect the dynamic characteristics of the patch membrane in your patch clamp?
I mean, first of all, I want to say that, of course, both of these systems have their good use and are useful, right? So this system is good for measuring tension sensitivity in cells, in living systems. You don't need protein purification for it, versus systems where you use a bilayer are more bottom up, and you can really determine, of course, lipid composition. Of course, the nature of the cytoskeleton changes the response of the membrane to pressure. But because our system adapts to it, it clamps tension to whatever value you want, right? So you might end up with a different curvature, right? But what matters is that you set it to the tension that you ultimately want to deliver.
So you mean the dynamic response may be affected by the cytoskeleton?
Not
the steady state. But
not the dynamic response. No. So if you perform measurements where you use this on live cells and you add agents that, for example, destruct the cytoskeleton, the response of the ion channel is not changed at the same tension values.
Jorg, wonderful talk. Thank you very much. I have two questions. You clearly showed the difference in the tension sensitivity between Piezo 1 and the Piezo 2. So far as I remember, also there is a difference in the activation and deactivation kinetics between Piezo 1 and Piezo 2, right? So these two factors are related to each other?
So there's no difference in the activation kinetics. Both ion channels open within less than a millisecond. So faster than we can or anybody can resolve.
Okay.
They do differ, as I mentioned, in the inactivation kinetics, and that was already shown initially by Bertrand Coste in the first publication that described piezo ion channels. But as I said, there are many other factors that heavily modulate inactivation kinetics. That's why, for me, it never made sense why you would need two different proteins for that.
Second question, short question, but quite important for me. Lipid bilayer is affected by temperature a lot. So my question is, your difference in the tension sensitivity should affect the temperature sensitivity of the two ion channel Piezo 1 and Piezo 2. Is that correct?
Yeah, so we have not tested this, but Slav Bagriantsev's lab at Yale a number of years ago, this must be 5-6 years ago, had a really beautiful publication where they investigated closely the temperature sensitivity of Piezo 1 and Piezo 2. They found that one of them indeed has an appreciable temperature sensitivity. But you as a trip channel guy, you might not be so impressed by it. The Q10 is about, if I remember correctly, only 3.5 or so. So it's far from what you would consider highly temperature sensitive. And importantly, of course, both proteins are not activated by temperature.
Okay.
They're modulated by temperature.
Thank you.
Let me ask a brief question. Yes, thank you very much for a wonderful talk. You mentioned about the curvature from lateral side, the view from the lateral side across the membrane. But if you look at the top perpendicular axis, a blade looks like a curved structure in 120 degrees. And I wonder the significance of curvature for each blade for sensitivity to the membrane stretch.
Yeah.
And also I wonder the trimer makes more sense rather than, for example, tetramer architecture for sensing the membrane tension.
Yeah, so to your first question, I should have maybe said this. We are measuring or capturing these images of the membrane inside the patch pipette under an angle that is as shallow as possible. And what we sort of have achieved is an angle of 11 or 12 degrees only. And this is not perfect. Ideally, you would want to go down to 0 degrees to really see the membrane perfectly perpendicular. But we've done an error calculation, and so the error that you get from basically looking at the projection of an object that is at an 11-degree angle versus 0-degree angle is less than 3%. So this is less than the experimental error that we get. So, the error is there, we are aware of it, but it's not a major contributor. The second question, piezos are trimers. There's no obvious reason that we know for why they are trimers, versus tetramers or dimers. There's no clear sort of energetic reason that I can think of that could explain that.
Thank you very much.
Unless there's any more questions, I'll probably close the session. I want you all to thank all the speakers in this session, and thanks to everyone, and you can run off and get your tea, pop to loo, do what you need to do.